28-year-old Jeffrey Robertson was found dead in his Mountain Brook home late Wednesday night. His coworkers reported Jeffrey missing after he had not shown up for work or answered any calls or texts since early Monday. The victim had a gunshot wound to the chest and several broken ribs, indicating signs of a struggle. Broken furniture and glass were found all over the room, along with a blood trail leading away from the body and out the door. Crime scene investigators collected blood evidence from the trail. The victim's DNA and blood samples were also collected and sent to forensic biology at the Alabama Department of Forensic Sciences in Hoover. Police questioned Robertson's close friends and discovered that he had been dating a girl named Lily Davidson. After further investigation, the police found that Lily Davidson had just escaped from an abusive relationship with a guy named Jackson Hartzell. The police brought him in for questioning, and detectives soon discovered that Jackson was the killer due to his blood being a perfect match to the blood trail found in Robertson's house. Forensic biologists examined the evidence using a promising analytical technique known as flow cytometry. Expert forensic biologist Dr. Brian Perry is here to further explain how this technique was used to help solve the crime.
So flow cytometry is a pretty interesting analytical technique. Essentially, it's a way for us to separate cells and then analyze them one at a time. As you can see in this diagram of a typical flow cytometry instrument to my left, we have a column, and we're basically going to push the cells through that column so they go through one at a time. Now down here we have some lasers. They shine light at a very specific wavelength, and they're going to hit the cells as they all pass through the laser's pathway. Lasers contain energy. They contain light. Light contains energy. That energy can excite the cells in what is called absorbance. When excited, those cells will then soon return to their ground state in a process called fluorescence. This is important because the things in the cell that are causing the fluorescence, they emit light as part of their fluorescence at a very specific wavelength. This wavelength is then taken through a series of optical wires to the instrument where we read it. That's right, we're reading the fluorescence. So what we're using flow cytometry for in these forensic studies are a couple of things. One, we're going to analyze a blood sample to see if it matches, which of the suspects that it matches. Essentially, your red blood cells contain antigens that bind to them. This is, as a matter of fact, this is what determines your blood type, if you're type A, type B, type O, whatever. We have here in this table, you'll see listed the three types of the antigens, A, B, and H. In flow cytometry, we can use antibodies to bind each of these. The antibodies fluoresce at different wavelengths, and therefore flow cytometry can record the fluorescence of each antibody. This tells us the ratio of antigen A to antigen B to antigen H. Antigen H is always a very small amount. Notably, each person's blood contains a different ratio. So we take a blood sample from the blood trail that we found at the scene, and a blood sample from each of the suspects. Whichever ones match, that identifies our suspect. There's another way we can use flow cytometry though. When you die, your DNA starts to decompose. We can see this image here shows how the helix begins to just blacken and tar and get all messy. This kills the cells. Importantly, this happens at a steady rate for about the first 3 days after you die. Now, we're going to use a molecule called propydium iodide, shown here. It can enter and bind to the dead DNA, but the live cells, their membranes keep it out. So we're going to basically stain the dead cells with this molecule, propydium iodide. Propydium iodide, it fluoresces at about 617 nanometers, so we're going to send all of these cells through the flow cytometer, and basically the amount of cells that are dead, as we can tell from how much fluorescence we detect, that tells us how long our person has been dead. And this is important because we can compare this to the alibis of people. So say we determine that the person died at 7 p.m. on Monday night. If the alibi doesn't match up, hey, that's an important clue that person might be our suspect.
Flow cytometry is an effective method for identifying the postmortem interval based on amount of DNA degradation and identifying a person from red blood cell antigen ratios. It is more accurate than vitreous humor technique of analyzing the postmortem interval. Vitreous humor is gelatinous liquid in the eye that is extracted during autopsy, Potassium and urea levels contained in the vitreous humor give information about the postmortem interval. However, the rate of decomposition of these levels is highly dependent upon environmental conditions such as temperature. The method's variability makes its accuracy questionable. Flow cytometry is preferred because DNA degradation occurs at a steady rate, although the rate may vary from person to person. Overall, flow cytometry is more sensitive, faster, and easy compared to other methods of investigation. Its sensitivity is based on the fact that a small sample size can be analyzed and that every cell in that sample can be studied. This method is faster because it has a shorter assay time. Hundreds of thousands of cells can be analyzed per hour. These qualities are preferred to reduce the number of pending cases in court. However, this method does have some drawbacks. Flow cytometry requires a cell sample that has a reasonably slow DNA degradation. There is also variation between instruments, meaning that each instrument would have to have its own validated methods. The same method from one instrument could not be applied to another. More studies with this application on different cases of death are required to establish a universally accepted rate of DNA degradation to avoid error in judgment.
In addition to the applications of flow cytometry in forensic science, we can take advantage of the fluorescent capabilities in a variety of ways. For example, infections can be detected, such as staph infection caused by Staphylococcus aureus cells. Essentially, if you collect a cell sample and then amplify any staph cells that may be in that sample with bovine serum albumin, we can then bind those staph cells with vancomycin and pig immunoglobulin G. Both of these fluoresce, and we can run flow cytometry to detect that fluorescence. Essentially, the higher the intensity of that fluorescence is, the more staph cells you have, and therefore the greater chance that you are infected. Similarly, we can detect certain cell anomalies that are present in cells using flow cytometry. For example, the disease human African trypanosomiasis, or the African sleeping sickness, is a good case study for this. It is transferred by tsetse fly bites from flies that have been infected by Trypanosoma brucei. How this works is they cause small inhibitors of glucose uptake and regulation to enter the body. We can use flow cytometry to screen for and detect these inhibitors. Once again, we simply attach a fluorescent antibody to that will bind to the inhibitors, and then use flow cytometry to detect the fluorescence. This is our video. These are references that we use to create this wonderful material. If you like it, hit the subscribe button to follow us for more exciting content.